Hello everyone, my name is Yusuf and this is our first IMAT Biology lecture. Before going into any topic related to IMAT, we will be discussing first why that chapter or that specific topic is important, what topics we have to study and what topics we have to leave, and what are the strategies to tackle the chapter according to IMAT syllabus, and what are the past paper MCQs that are most important that will be coming in the upcoming exams, and we will be discussing all the tips and tricks that you can use in that specific chapter. After the chapter lecture is done, there will be a written test, a quiz you have to attempt. Then you will be given all the premium past papers for TOLK, for a BMAT and IMAT because they are really relevant to our upcoming exams. And then after that, you will be studying the premium notes that will be given to you. So after that, your preparation will be done from 360-degree. Like you won't need anything after all of these things. These are all things that you need to clear out your concepts and you complete your practice material. This is all the material that you will need to score the highest marks possible in IMAT. And I will be teaching you step by step at every single point about how to tackle that chapter, how to tackle the MCQs, and how you can maximize your concept in that specific topic. So first chapter is about the chemistry of life. Actually, this chapter is one of the most important chapter in IMAT. The reason is because this chapter doesn't contain so many MCQs in IMAT. Like there might be 1 or 2 MCQs every year, but that is not why this chapter is important. This chapter is important because this chapter is the base of biology. Like without understanding this chapter, understanding the further chapters that we'll be studying is going to be really difficult. Because if you don't know the basics of carbohydrates, for lipids and amino acids and nucleic acids, then studying about organelles, cell and bioenergetics and respiration, those kind of reactions and processes, understanding them is going to be really difficult. So that's why understanding this chapter is so important, because if you have a weak base, then the building that you will be stacking upon that base is going to be falling apart if the base is not strong. So the first topic that this chapter contains is water and its properties. It is like a medium yield topic because there are going to be 0 or 1 questions from this. Like in 2024 there was 1 MCQ from this topic, and then we have carbohydrates. It's also medium yield. There are going to be 0 or 1 MCQs. It was tested in 2024. Then we have lipids and proteins and nucleic acids. So this is the list of questions that you have to study. You can take a screenshot and then we will move to our chapters. Okay. So the first thing that you have to know is what is biochemistry. So biochemistry is the branch of biology that deals with the chemical processes that are occurring in your living cells. The first thing that you have to know is that there are 92 naturally occurring elements in all living organisms, and out of those 92 there are 25 elements that are found in living beings, and out of 25 elements 16 bioelements are present in human cells. And out of those 16 bioelements, 6 are major and 10 are minor bioelements. So you have to memorize the 6 major elements that are really easy to memorize. So their mnemonic is called OCHANCAP: oxygen, carbon, hydrogen, nitrogen, calcium and phosphorus. You can like use any mnemonic you want to. Like I think that this mnemonic is really easy. So the mnemonic is again OCHANCAP. Oxygen actually contains 65%, and then carbon contains 18%, hydrogen contains 10%, nitrogen contains 3%, calcium contains 2%, and phosphorus contains 1%. So that's how the major elements contains 99% of your cell. So now you understand what are the elements that are present in your body. So these are the elements that are the most important. You have to try to memorize them. Oxygen contains 65, carbon contains 18, hydrogen contains 10, nitrogen 3, calcium 2, phosphorus 1. Easy peasy lemon squeezy. So the other thing that you have to know is from minor elements, iron is one of the most important. Why iron is the most important? Because iron is present in all of the living organisms. Like all the living organisms require iron to survive. Without iron, no living organism can survive. So that is why that is the most essential element that is present in minor bioelements. So you have to memorize this point. So if you have memorized this part, elements, then let's move on to our chemical components. So what are the chemical components that are present in a human cell? So you have to know what chemical components are most abundant and what chemical components are least abundant in a human cell. So you have to memorize it as W, P, C, L, D, R. Like you can use any molecule you want to. W is water, P is protein, C is carbs, L is lipid, D is DNA, and R is RNA. So as we move from top to bottom, the chemical components are becoming less and less in our body, because water is the most abundant chemical component in our body right now. Water comprises about 70%. 70% of our cells contains water. Okay, so we are talking about cell here, not our whole body. Like our cell contains 70% water. And if we are talking about protein, then it is 18%. And if we are talking about carbohydrate, that is 4%. If we are talking about lipid, that is 3%. DNA is 0.25%. And RNA is 1.1%. So you don't have to memorize the percentages here. You can just memorize what is most abundant. Like water is the most abundant component in our cells. Then we have protein, then carbohydrate, then lipid, then DNA, and then RNA. So if you memorize these, then you are good to go. You don't have to memorize the percentage, because the percentage is, they won't be asking about a percentage in IMAT. Yes, there is a tricky question here. You have to be able to answer that which of the following is the most abundant organic compound? Then the answer will be proteins. If the question is which of the following is the most abundant chemical component, then the answer is going to be water. So you have to be really careful about what they are asking about. Like what is the most abundant bioelement in your body? Then it is going to be oxygen. And if they are talking about a component, then it is water. And if they are talking about organic compound, then it is protein, because water is inorganic and protein is organic. So the most abundant component that is organic in our cells is protein, then it is carb, then it is lipid and DNA, and then so on. So you have to understand these kind of questions if you want to excel in IMAT biology. So let's move on to our next topic, that is about water. So water is like our first topic. Water is the universal biological solvent, and it is the most abundant chemical component in our body, because our body is made up of 60 to 90%, as we have discussed right now. Like 20% of bones are made up of water, right? 80 to 90% of your brain is made up of water. Fun fact, 99% of jellyfish is water. Like jellyfish is 99% made of water. So there are organisms there that has so much water into their body. So water is the molecule of life. Without water, the life is impossible. So water is a polar molecules because oxygen is strongly electronegative and hydrogen is partially positive. So because of these two ends, the water is a polar molecule. So let me draw the water molecule for you, so you can understand it clearly. Water molecules contains oxygen and two hydrogen molecules. And if I draw another water molecule here and another one here. So first of all, you have to understand what is a hydrogen bond and what is a covalent bond. If you can see the bond between oxygen and hydrogen within a water molecule, just like that, has covalent bond. So within a water molecule, the bond is called covalent bond. And between water molecules, the bond is called hydrogen bond. So hydrogen bond is actually between water molecules. So whenever you boil water, then the bond that breaks is called hydrogen bond. If the covalent bond breaks, then that means the water molecule has been disintegrated. It is not water anymore. It is just oxygen and hydrogen. So you have to keep these kind of things in mind when you are studying water, because so many students get this thing wrong. So I thought that maybe I should be discussing this topic. So there are 4 essential properties that are mostly discussed in IMAT Biology. The first is about cohesion and surface tension. This MCQ was asked recently. Like from these 4 topics, IMAT has been asking a lot of questions. So you have to know these 4 aspects of water molecules really deeply. So the first is cohesion and surface tension. So what is cohesion and adhesion? So cohesion is a force of attraction between same molecules, and adhesion is a force of attraction between water and different molecules. For example, you have studied how water is transported in plants. So water forms a long chain like 1, 2, 3, 4 water molecules, and they are attached to each other through hydrogen bond. That is actually cohesion. And water molecules also attach with the xylem cells, and they climb up the xylem tissue like that slowly. The attraction between water and xylem tissue is called adhesion. So water molecules has 2 kind of forces, cohesion and adhesion. So in this way, water can move from one part to another part very easily. So our next topic is high specific heat capacity. So what is heat capacity? Heat capacity is the amount of heat required to raise the temperature of water through 1 degree Celsius. Like you are providing heat to water and provide again and again and again until the temperature of water increases by 1 degree Celsius. Then that amount of heat is called specific heat capacity. So specific heat capacity is really important, because whenever water molecules are bound together, the heat capacity remains the most crucial thing, because water resists temperature changes because of heat capacity. For example, whenever you give energy to water molecules and you boil water, then specific amount of heat is given, and to break the hydrogen bonds you have to give a lot amount of energy, like a lot. That's why water act as a thermal stabilizer. For example, the cities and the countries that are close to water are not very cold and not really hot. They are like Mediterranean cities, and just like India, just like Pakistan, they have all the 4 seasons because they are really close to the sea, because sea acts as a thermal stabilizer. What it does is that if there is a really high temperature, then it will absorb the heat, and if there is very low temperature, then it will also stabilize its temperature. So water act as a thermal stabilizer. So heat does not increase a lot and decrease a lot. Like it can stabilize the temperature in its surrounding. So the next topic is universal solvent. Water is a universal solvent. It can dissolve anything, and the reason is because it is a polar molecule. as we know that water is a polar molecule, so for example, if there is a molecule that is partially positive charged, for example, if we have sodium, what will happen? It will be attracted by the partially negatively charged oxygen atom. If we have chloride ion, then it will be attracted by the partially positive charged hydrogen atom. So if positive or negative charged atom is mixed in molecule, then they will be soluble. And another example is about non-polar molecules. Like we have discussed polar molecule like sodium chloride. What about non-polar molecules? Non-polar molecules will be dispersed in water. There is very high chances that non-polar molecules will disperse, and they will also dissolve into water. That doesn't mean they will form any kind of bond, but they will just dissolve in it, because there is a difference here. Whenever a bond is formed, the covalent bond of water will break, but whenever something is dissolved in water, then hydrogen bond will break. The same is for non-polar molecules. When a non-polar molecule is dissolved in water, that doesn't mean that non-polar molecule will form a chemical bond with water or it will react with water, but rather than that, it will just break the hydrogen bonds that are present, and it will disperse between water molecules. Like it will dive and swim between water molecules, not actually forming any kind of bond. I think you understood my point. The next point is ice is less dense than water. You have to know that, like, why ice is less dense than water. Like, this is the most common question that's been asked in IMAT again and again. So you have to first know what is mass and what is density. So density is actually mass per unit volume. If something has much more volume, then its density will decrease. So any molecule that has more volume, its density will be low. The first example is going to be about, obviously, water. You have noticed that if you store water in a refrigerator, then after a few hours, the ice is increased in volume after some time. Like you can see a little bump into the ice. The reason is because you can see the water molecules. They are opening and closing just like this. You can see like they will open and then close and open and close and open and close. But whenever you freeze water molecules, they will just stop moving and keep their position like this. They will not shrink the hydrogen atoms, but they will freeze into one location. So that's how their volume will increase after freezing. The fun fact is, everything that is freezed, its volume will decrease. For example, if you lower the temperature of any molecule, any chemical component in the whole world, its volume will decrease, except water. If you freeze water, then its volume will increase rather than decreasing. So it is an exception to chemistry rules, water. So water's volume will increase and its density will decrease. That's why ice is less dense than water, because its volume increases and its density decreases. So that is the reason that ice floats in water, because if something is less dense, then obviously it will be floating on something that is more dense. So water is more dense and ice is less dense, so ice floats on water. So that was from our first topic. Let's move on to our next topic, that is about carbohydrates. So carbohydrates are literally called hydrated carbons. Hydrated carbon means that it contains carbon, hydrogen and oxygen. So these are three primary atoms that are present in all of the carbohydrates. So carbohydrates do have only three major roles. Like the first is they store energy, they give structural support, and they act for cellular communication, just like glycoprotein and glycolipids we will be discussing in our next chapter. So carbohydrates have three types. The first is monosaccharide, then we have oligosaccharides, and then we have polysaccharides. So let's discuss our first topic, that is monosaccharides. So monosaccharides are the basic carbohydrates. They contain 3 to 7 carbon atoms in a monosaccharide. Like there could be minimum 3 carbon atoms in a monosaccharide. The most common examples are glucose, fructose, galactose, ribose, and deoxyribose. Like these are the most important monosaccharides that are present. So glucose and fructose contains 6 carbons, galactose also contains 6, ribose and deoxyribose contains 5 carbon atoms. There are so many other examples. You can know that there is glyceraldehyde, there is dihydroxyacetone. We will be discussing that example. Let's discuss that first, to be honest. So first example is glyceraldehyde, that is a 3 carbon monosaccharide, C, C and C. Glyceraldehyde contains aldehyde group, and other is dihydroxyacetone. It contains ketonic group like this. So glyceraldehyde contains aldehyde group and dihydroxyacetone contains keto group. So these are the simplest monosaccharides that are present in our society. This is glyceraldehyde, and then we have dihydroxyacetone. So glyceraldehyde and dihydroxyacetone are the most basic monosaccharides, and they are present in photosynthesis, in respiration, and they are really important molecules in our daily life. So let's go with glucose, fructose and galactose. So glucose is also, as you can see, aldohexose. It contains 6 carbons and it contains aldo, and it contains, fructose contains keto group. Galactose also contains aldo group. So what is the difference between glucose and galactose? If they have same formula and if they are both aldo sugars, then what is the difference between them? So you have to know that there are actually 3 molecules that are really same. They are glucose. Let me write it down for you. Glucose. The next is mannose, and the third is galactose. So these 3 molecules are really same: glucose, mannose and galactose. How are they same? As you can see, the glucose molecule contains OH group on 3rd carbon, while mannose contains carbon number 2 and galactose contains carbon number 4. That is the only difference. Like mannose has OH group in the opposite side in carbon number 2, galactose has OH group on the opposite side in carbon number 4, and glucose has on carbon number 3. If you still can't understand what I'm saying, let me draw it down for you. Glucose contains 6 carbon, 1, 2, 3, 4, 5 and 6. And it contains OH group like this: OH, OH, OH and OH. As you can see on carbon number 1, 2 and 3, the OH group is on the left side. But for mannose, it is on the left side on carbon number 2, and for galactose, it is on the left side on carbon number 4. So this is a really minor detail that you have to remember when you are memorizing glucose, mannose and galactose, because they have the same category, they have the same formula, but they have just a little bit different structural formula. Next is about ribose and deoxyribose. Both are aldopentose, but the one difference that they have is that ribose contains one more oxygen than deoxyribose. Like the name is telling you everything. Deoxy. Deoxy means that there is one less oxygen in deoxyribose. So these are the most important carbohydrates that you have to memorize in monosaccharides because they are essentially the most important things. Let's see the key features here. Oh, one thing that I forgot to mention here is that what are ring structure and what are straight structures. So let's discuss this topic too, because this is actually a really important topic to discuss. I can't find any place to write it down. Like, where should I be writing it? Okay, so we can write it down here. So there are actually two forms, two structures. The first is straight chain structure and the other is ring structure. Straight chain structure is when the molecule is present in dry condition, and whenever the molecule is dipped in water, like in aqueous condition, it will form a ring structure. So glucose, fructose, ribose, deoxyribose, all of the monosaccharides do form straight chain structures, and they also have ring structures. So they have both ring structures and straight chain structures. So if they have ring structure, then what kind of structures they will have? They can have Furanose or Pyranose. Sorry about that. Okay, so they can have Furanose or Pyranose. Furanose and like glucopyranose, ribofuranose, you guys have already memorized these names, but you didn't know where they came from. So they are two types of ring structure. First is Furanose and other is Pyranose. Furanose contains 5 corners. It is a 5-membered ring, and Pyranose contains 6 corners. It is a 6-membered ring. So you have to memorize which kind of molecules will form Furanose and which type of molecules will form Pyranose. So all pentoses and hexoses, will form Furanose, while only aldohexoses will form Pyranose. The aldohexose example is glucose. Glucose forms glucopyranose, and other hexose is fructose. So that's why fructose forms fructofuranose, right? And all the other pentoses also form furanose, like ribose, deoxyribose. So that's how furanose and pyranose are diagnosed. Like if you are asked which of the following forms furanose structure, which of the following forms pyranose structure, so now you know that all pentoses and hexoses form Furanose structure and all aldohexoses form Pyranose. Like glucose, mannose and galactose, they form Pyranose ring structure. So let's come back to our topic. Okay, as you can see, the glucose contains 6 corners: 1, 2, 3, 4, 5 and 6. So these 6 corners are telling us that glucose forms glucopyranose, and then fructose, ribose and deoxyribose, all of them contains 5 corners, so they are forming Furanose structure. So that's how you can diagnose which of the following contains which type of ring structure. I think I have cleared all the concepts and all the things that we need for IMAT monosaccharides. So let's move on to our next topic, that is disaccharide. Disaccharide is actually oligosaccharide. So oligosaccharides contains monosaccharides from 2 to 10. Like if there are 2 monosaccharides or 9 monosaccharides, then it is a disaccharides. So we will be discussing disaccharides for today because they are the only questions that are asked in IMAT. So the first most important disaccharide is maltose, then sucrose and lactose. So how disaccharide is actually formed? There are two types of bonds in biological molecules. The first is, you have to know, dehydration synthesis, that is called so disaccharide is formed by a reaction called condensation. So disaccharide is formed by a reaction called condensation. Condensation is also called dehydration synthesis. Why is it called dehydration synthesis? The reason is simple, because it causes dehydration to cause or to synthesize a molecule. Let me give you an example. For example, if you have a glucose molecule like this, and it contains OH group, and another glucose molecule like this. So to form a bond between these two molecules, what will happen? The OH group from first glucose and O group from second glucose, they will be removed and a water will be formed, H2O, and a bond will be formed. As you can see, these two glucose molecules will form a bond using this hydrogen, and this reaction is called condensation reaction or dehydration synthesis, because a water molecule has been removed here. So what if you reverse this reaction? Like if you break this bond and move backward, it will be called hydrolysis. What will you call this reaction? Hydrolysis. So hydrolysis actually mean forming or breaking a bond using water molecules. Like lysis means breaking something or cutting something into half, and hydro means water. So breaking a bond between two molecules utilizing water, using water, is called hydrolysis. For example, if you want to break this bond, you will have the water molecule inside here. So water molecule will go there and the two glucose molecules will be formed again. So this is called hydrolysis. So hydrolysis is the reverse of condensation reaction, that is also called dehydration synthesis. I think the concept is clear here. So that's how disaccharides or oligosaccharides are formed. So there is another concept that you have to remember, like how many water molecules are released during condensation reaction or dehydration synthesis. If I ask how many water molecules will be released if 3 glucose molecules join together to form an oligosaccharide, then the answer will be 2 water molecules. Why is that? Because if you have 1 glucose molecule, 2 glucose molecule and 3 glucose molecules, they will have 2 bonds between them and 2 water molecules will be released during that reaction. So the formula is number of monosaccharides that joins together minus 1. So the shortcut formula is number of water molecules equals monosaccharides minus 1. Like the number of monosaccharides that are forming the bond minus 1. Like if there are 10 monosaccharides that formed bonds together by condensation, then there will be 9 bonds and 9 water molecules will be released in that reaction. So the next 3 molecules that you have to memorize here is maltose, sucrose, and lactose. Like maltose contains two glucose molecules, sucrose contains glucose and fructose, and lactose contains glucose and galactose. So there contains alpha 1,4 glycosidic bond, alpha beta 1,2 glycosidic bond, or beta 1,4 glycosidic bond. So what are these bonds? What do they mean, and how can we recognize what is alpha, what is beta? So you have to understand the glucose molecule structure first. So to understand alpha and beta, let me draw some diagrams here. So we have studied two types of structures. The first was ringed structure and the other was straight chain structure, right? So if a molecule contains OH group and the OH group on the carbon is on the downside, and if the OH group is on the upward side, then we can recognize if it's alpha or if it's beta molecule. So the simple question here is, if the OH group at carbon number 1, for example, let's say this is the carbon number 1 of glucose, and this is also another molecule, and it's also the carbon number 1 because we have less space. So if the OH group is downward, then it will be called as alpha sugar. And if the OH group is on the upper side, then it will be called beta sugar. So that's how alpha and beta sugar can be recognized. What about the straight chain structure? Like if there is a structure that is straight chain, then how can we recognize it? If the OH group is on the right side, then it is called alpha, and if it is on the left side, then it is called beta. If the carbon number 1 contains OH group on the right side, then it is alpha, and if it contains OH group on the left side, then it is called beta. Oh, there is something else that you have to know here that is really interesting, like how a straight chain structure is changed into a ring structure. Imagine this is a straight chain structure like this. You don't have to do anything. If you have memorized a straight chain structure for a molecules, you just have to like bend the straight chain structure and connect the last two carbons like this, and then you will have a ring structure. For example, for glucose molecules there are 6 atoms, so you just have to take this carbon and this carbon and connect them with each other. If the OH group is on the right side, then it will be alpha because it will eventually come on the downside if we turn the whole molecule into a ring. As you can see, the OH will be on the downside, and if it is beta, the OH will be on the upper side. If we turn the ring structure into, if you turn the straight chain structure into a ring structure. I think like we have discussed mostly everything that is there for disaccharides. So let's come back to our topic. So now we know what is maltose, what is sucrose, and what is lactose. So you have to memorize that maltose forms 1,4 glycosidic linkage. Alpha means that the OH group is on the downside, and beta means that the OH is on the upper side of carbon number 1. What about 1 and 4, 1 and 2, 1 and 4? What it actually means is that 1 and 4 means the bond is between carbon number 1 and carbon number 4. 1 and 2 means the bond is between carbon number 1 and carbon number 2 of the other molecules. Because glucose carbon number 1 will form bond with carbon number 2 of fructose. Because fructose contains 5 ring structure, 5 cornered ring structure, and glucose contains 6 cornered ring structure. That's why they forms 1, 2. And as they are both glucose molecules, both of them contain 6 ringed structure, they will form 1, 4. Glucose and galactose also contains 6 cornered ring structure, they will form 1, 4 glycosidic bond. So that's how you can memorize most important disaccharides. Like you have to memorize what are the monomers that are used in disaccharide, what kind of bond they are forming, and if they are alpha or beta. That's really important. You have to know these kind of facts, these kind of concepts, and if you understand these concepts, then doing these kind of MCQs is going to be a piece of cake, believe me, because I think I have made them really easy, and we have discussed everything that there is about disaccharide. So let's move on to our last topic, that is polysaccharide. So polysaccharides are chain of monosaccharides. Like if so many monosaccharides, like large number of monosaccharides, more than 10 monosaccharides connect with each other, then they will form polysaccharides. So what are the most important polysaccharides? The first is starch, then glycogen, then cellulose, and then we have chitin. Okay, so first thing you have to know is that starch is made up of glucose molecules. It contains alpha glycosidic bond. Its primary function is energy storage, and it is mostly present in plants. The next molecule is glycogen. Glycogen contains both alpha 1,4 and alpha 1,6 bonds. If we are making glycogen, then it contains bonds like this, alpha 1,4, alpha 1,6. Like they will form bonds from ahead and above. So that's how a glycogen molecule is formed. It contains both alpha 1,4 and alpha 1,6 bonds. That's why it is highly branched. As you can see, there are so many branches in a glycogen molecule. So that's why glycogen molecule is really important. Then you have cellulose. Cellulose is made up of glucose molecules. It contains beta 1,4 glycosidic linkage. And then you have chitin. It is made up of N-acetyl glucosamine. Actually, N-acetyl glucosamine. So chitin is made up of N-acetyl glucosamine. N-acetyl glucosamine is actually nothing but a modified form of glucose. It contains nitrogen, and chitin is formed from this molecule called N-acetyl glucosamine. You have to memorize these things, they are really important. So let's move to our detail to these polysaccharides. The first is about starch. Starch contains two types. The first is amylose and the other is amylopectin. So amylose and amylopectin are really important to memorize. Before we study amylose and amylopectin, I have to discuss an MCQs that was asked recently, like which of the following is a homopolysaccharide and which of the following is heteropolysaccharide. Last year this question was asked. So the polysaccharides that are written here are homopolysaccharides. Like all four of these are homopolysaccharides. And if you are asking about hetero, then their examples are agar, pectin, etc. Like they are also polysaccharides, but they are heteropolysaccharides. What are heteropolysaccharides and what are homopolysaccharides? Homopolysaccharides are formed by a single monomer. Like starch is made up of only glucose, glycogen is only made up of glucose, cellulose is only made up of glucose, chitin is only made up of N-acetyl glucosamine. While heteropolysaccharides like agar and pectin and etc., they are not made up of only one monomer. They are formed by different kind of monomers. You don't have to go into the detail. You only have to memorize most important homopolysaccharides. So they are homopolysaccharides. This was asked in 2025. So you have to memorize this. So starch has two types, amylose and amylopectin. So first of all, what is amylose? First, amylose is like less branched or unbranched type of starch. And if you are talking about amylopectin, it is branched type of starch. Let's talk about the next difference. Amylose is soluble in water, in hot water, okay, not in simple water. But amylopectin is insoluble even in hot water. Like amylopectin is never soluble no matter what you do. So that's the difference between starch, two types. The first is amylose and the other is amylopectin. Both are types of starch. Amylose is unbranched and it is soluble in hot water, while amylopectin is branched and it is insoluble in hot water. The next thing that you have to keep in mind here is that there are two things like amylopectin and glycogen. They are really alike because both are branched. Amylopectin is branched and glycogen is also branched. Like both of them are branched molecules. So they are really alike from each other. But there is a bit of difference that glycogen is highly branched, like it contains really high amount of branches, while amylopectin doesn't contains really high amount of branches, it contains only a small amount of branches. So that was about starch and then glycogen. Okay, so next thing that you have to know is that starch doesn't break directly into glucose. What does it mean? Like starch first breaks into maltose. We have already studied that maltose is a disaccharide containing 2 glucose molecules, and then maltose will break into glucose molecules. Most of the students make this mistake a lot of times. So you have to know that starch first break down into maltose and then maltose breaks down into glucose. So glucose is the basic structure of starch, like this. So glucose forms together to form maltose, and then maltose forms together to form starch. So you have to keep this point in mind, because in IMAT these kind of questions are really common. Like they will ask you, if we break starch, then the immediate product will be, and students are like, oh, we have studied this, it is a polysaccharide, it is formed of glucose, so the answer is going to be glucose. No, it is not going to be glucose, it is going to be maltose, because they have asked about the immediate product. If they are asking about the final product, like if you break starch, if you do hydrolysis of starch, what is going to be the final product, then it will be, then the answer will be glucose. But the answer for the first or immediate product will be maltose. We have discussed a lot of things today here, so let's revise everything we just did. Before that, let's do some MCQs. Like if I ask you that if tricondensation reaction occurs, then how many water molecules will be released? Think for a moment. Yeah, if it is a tricondensation reaction, then it will form 2 water molecules, because for tricondensation reaction there will be 3 molecules of glucose and glucose, and 3 molecules will form 2 bonds and 2 water molecules will be released. So you can also find out how many molecules are attached using the number of molecules released. For example, if they tell you that 2 water molecules are released, then how many glucose molecules are present in that specific polysaccharide, then you will be think about, like if there are 2 water molecules, then that means that there will be 3 glucose molecules. So that's how you can also see it from the other side. There is another MCQs that we discuss here. Let me write it down somewhere. We have filled every single place. We don't even have any place to write down MCQs. Okay, so that's all the MCQs that we had to discuss here. We will be discussing more number of MCQs in practice. So that's all from my side. I think we have discussed everything. Let's meet into our next lecture. Bye bye.